Thyroid gland is located just below the larynx, which is a part on endocrine system. It has two lobes situated on either side of the trachea. Thyroid gland consists of numerous spherical hollow sacs called thyroid follicles, which are lined with a simple cuboidal epithelium called follicular cells, which interiorly contain colloids as protein-rich fluid. Thyroid follicles synthesize majority of thyroxine, T4, as the principal thyroid hormone, which then converts into triiodothyronine, T3, in the target cells. These hormones are very important for determining the basal metabolic rate, which provides proper growth and development for individuals. In addition, thyroid gland also contains parafollicular cells that secrete a hormone called calcitonin, which helps to raise the blood calcium ions concentration. The thyroid follicular cells actively accumulate iodide from the blood and store it within the follicle. Once inside the colloid, iodide was oxidized by hydrogen peroxide facilitated by peroxidase enzyme. Oxidized iodide bound with specific amino acid tyrosine of a protein called thyroglobulin. The bound on 1-iodine to tyrosine produces monoiodotyrosine, MIT, and 2-iodine to tyrosine produces diiodotyrosine, DIT. Within the colloid, both MIT and DIT undergo structural modification by various enzymes, in order to couple them together. When two DIT molecules that are appropriately modified, they are ready to be coupled, giving a molecule of tetraiodothyronine or thyroxine, abbreviate T4. And the combination of one MIT with one DIT forming triiodothyronine, abbreviate T3. Upon stimulation by TSH receptor on thyroid follicular cells inducing cellular uptake of a small volume of colloid, then hydrolyzes T3 and T4 from thyroglobulin to secrete the amount of free hormones into the blood. The carrier proteins have a higher affinity for T4 than for T3, that result the amount of unbound or free circulated T3 in the plasma is about 10 times greater than the amount of free T4. About 99.96% of the T4 in the blood is bound to carrier proteins, giving thyroid-binding globulin, which serves as a hormone reservoir. That's why it has to take a couple of weeks after thyroidectomy procedure for the symptoms of hypothyroidism to appear. Only the free T4 or T3 could enter the target cells, and once inside the cytoplasm, there are enzymatic reactions ready to convert T4 to T3. However, free T3 may enter the cell from the plasma directly. After that, it passes through some nonspecific binding protein in the cytoplasm that we called stepping stones process to enter the nucleus and binds with the thyroid hormone receptor, which is located inside. Once inside the nucleus, T3 binds to its receptor and alters the shape of the complex, forming a monomer structure, which could enable for removal of co-repressor proteins and recruitment of co-activator proteins that promote genetic transcription. However, T3 does not work alone, because the thyroid hormone response element on DNA has two half sites, and the thyroid hormone monomer binds to only one of the half sites. The other DNA half site has to binds with receptor for the 9-cis-retinoic acid as a vitamin A derivative. When two heteromonomers, one from thyroid hormone receptor and one from the 9-cis-retinoic acid receptor, bind to the two DNA half sites of the hormone response element, it form an IgA heterodimer. This would result in activation of genes to stimulate protein synthesis, promote maturation of the nervous system, increase the rate of cell respiration in most tissues of the body, and elevate the basal metabolic rate. Without thyroid hormone, thyroid receptors would recruit co-repressor proteins to inhibit the DNA hormone response element, so, the nuclear receptor proteins for thyroid hormone cannot stimulate genetic transcription until they bind to their ligands. That's it for the topic. If you find this video useful, please give us a like and don't forget to subscribe our channel. Thank you for watching.